Hi everyone, this is lecture 7, Patterns of Inheritance, and this comes from Concepts of Biology, chapter 8. So this kind of builds on what we were talking about the last couple lectures. So mitosis is a way of creating more cells when your body needs more cells. So for example, growth and development, for asexual reproduction, and then meiosis, even though the names are similar and it is another way of dividing cells, meiosis is specifically related to making more gametes, which are the reproductive cells like sperm or egg cells. And so that kind of ties into this lecture. So patterns of inheritance, in other words, genetics. So hopefully by the end of this section, you'll be able to explain the scientific reasons for the success of this guy called Mendel and his experiments. Hopefully describe the outcomes of different crosses of different organisms. Explain the relationship between genotypes and phenotypes. Use a Punnett square to kind of visualize how different crosses and matings produce different offspring. Explain Mendel's laws, and explain the purpose and methods of a test cross. So again, all of this will make more sense hopefully by the end of this lecture. So we can't really talk about genetics without talking about this guy called Gregor Mendel. So he's known as the father of genetics because he did some really important experiments that at the time were kind of overlooked for a while, but then people started realizing like, hey, this guy was onto something. And now, because of his work, we know that genes are carried on chromosomes, and those are the units of heredity that get like replicated, expressed, or mutated. So basically this guy noticed that things happen to produce offspring of, they reproduce in kind. So they produce offspring that look like similar to the parents, but sometimes some traits can like be similar, or some traits can kind of be hidden for a generation. And so he thought, I wonder why that is. And so he did this because he was a gardener, basically. So he was an Austrian monk, but he had a lot of spare time, and he also gardened. And so he used pea plants. So if you think about like, peas on a pod, they come from plants that have flowers. And so he observed like different traits about the flowers and about the peas. And so this was his model system. So model systems, we might have talked about the first lecture. They are a system with convenient characteristics that is used to study a specific biological phenomenon to gain understanding, and that can be applied to other systems. So for example, it's really hard to study, again, different things in like a long-lived species, like elephants, for example, or humans. It would take a long time to study, different generations of humans. So we use model systems to study a bunch of different concepts, and then that can kind of be applied to other systems. So his model system was the garden pea, and so he experimented with thousands of garden peas, and he showed that the traits that are passed on from parents to offspring, it happens in specific patterns. So by him tracking so many thousands of peas, he was like, well, this has to be something. This is something that gets carried on from parents to offspring. So he was kind of really lucky that he was studying peas. So the plants that produce, peas as we know them as like food, these plants are kind of, he was really lucky, honestly, because if he had studied a different kind of system or a different kind of traits, the patterns might not have showed as clearly as it did with these guys. So how did the peas help him? How did he get lucky studying these things? So one, pea plants are true breeding. So that means that if you let one of these plants self-fertilize, they'll keep producing the exact same kind of their own. So if you have a plant that has purple flowers and you let it self-fertilize because plants have reproductive parts of like both kind of like male and female. So they can self-fertilize. So if you let this plant self-fertilize, it'll carry on its own traits. But it's kind of cool too that you can physically manipulate, like I'm going to breed this one with this one by like rubbing the pollen onto the stamen of the other plant. So it helped him because these plants were true breeding. So like if you understand, you know, this plant, if I don't cross it with anything else, it'll only ever produce purple flowers. And then this plant will only ever produce white flowers if I only let it, you know, self-breed. So that kind of helped because there were not unexpected traits that would occur individually. So he was like, it means more if I cross these and something different happens, because that's due specifically to what I did. So the fact that these plants were true breeding, they only produced their own if they were allowed to self-fertilize, kind of really helped. These plants reproduce quickly, so like I mentioned, you know, humans, a long-lived species, it takes a long time to see, you know, traits in one organism, and then in the offspring it takes like a longer time. So you can't really study generations in as quick of a time. So the pea plants, they reproduce quickly, and several generations could be like evaluated over a short time. Large quantities of peas could be grown at once. So when we think about the peas in a pod, each pea is actually like an offspring. And then if you were to plant that pea, it's like a seed that will end up growing to become another plant. So by being able to like have one plant produce multiple peas, you're studying a lot of different offspring at once. Again, versus like adults where we only have, you know, years between children. Pea plants, you could see a lot of different offspring each generation. So because also he had such a number, he could have such a high number of offspring, he could also see like, okay, these patterns really mean something. If I'm counting thousands of these peas and there's a pattern for thousands of peas, then that means something. There is a pattern and it wasn't just chance. So he really got lucky. And he also happened to be studying things that had discontinuous variation. So some traits that we say have continuous variation means like there's a lot of variation and in a continuous way. There's a range of small differences among individuals in this characteristic. So for example, human height. So humans, you know, they're not just short, medium, tall. There's a continuum of the different heights you could be. So that's continuous variation, and that's kind of harder to study and see the patterns that he saw. So he happened to get lucky studying something that had discontinuous variation. So it's kind of just like groups. So it's either violet flowers or white flowers. It's either round seeds or wrinkly seeds. So he, studied discontinuous variation in these things, where you can really easily distinguish like the different traits. It's either this or this. So when he was doing these different crosses, like I mentioned, you can kind of allow plants to self-fertilize because they have both reproductive structures, but you can manually transfer the pollen from one plant onto the ovum of another plant, and that crosses them. So for example, he wanted to see what happened when you crossed a plant with violet flowers or purple flowers with a plant that produces white flowers. And again, if you let them self-fertilize, they'll each produce, you know, one that looks exactly the same. But if you cross-fertilize them, he wanted to see how would that work? Like, what would happen? So when he did his crosses, he kind of had to come up with a naming system because that would help him, you know, keep track of the different generations. So the first generation where he physically crossed them was what we call the P generation. P stands for parental. So the P generation is the one that he actually crosses, you know, let me take the pollen of this purple flower and cross it with the one with the white flowers, and then we'll see what happens. So then those produce offspring, and so the offspring are all called the F generation. So that stands for filial or familial. And then we could just put the number on which generation that is. So F1 is the first filial generation. So those are the offspring of the cross that he did of the parents. So F1 and then F2 would be the next generation. So he crossed the P generation, purple and white. In the F1 generation, he found that they were all purple flowered. They all produced purple flowers. And he was like, oh, okay, that's interesting. What happened? What happened to the white flowers? Did it just disappear? Like, does it get erased? And then the purple is all that's there? And so he did this again. He actually crossed one of the F1 with another F1. Well, self-fertilized. So he was like, okay, what if we self-fertilize this one and see what happens in that offspring? So then that is the F2 generation. And when he did that, he found out that the offspring of these purple flowers had a mix of purple and white flowers. And he was like, huh, that's so interesting because the white disappeared. So that means that like the white color of the flower doesn't just get erased. It must stay there somehow, like hidden until the next generation. And so he did this, not just with one set of flowers, he did this with multiple. And so again, by reproducing so fast and having so many offspring, he counted in this generation 705 plants with the purple flowers and 224 plants with the white flowers. So it wasn't just random chance, like there was a lot of them. And he did this with other traits as well. And so he found the same kind of pattern with each of these traits. So if he was like, okay, that's weird, if it's just purple and white flowers, what if I just looked at something else? What if I looked at the seed shape? If the peas themselves were round versus wrinkled, what would that look like? So if I cross a plant that only gives round and then a plant that only gives wrinkled peas, what happens to the offspring in those peas? What do they look like? And he found the same thing. The first F1 generation was all round. And then the next generation had a mixture of round and wrinkled. So that trait, again, didn't just disappear. It was just kind of like hidden. And so he did that again with a different trait. So what about seed color, yellow versus green? What about pod shape? Like if it's kind of smooth versus like you could really, like bumpy, you could see the different peas inside the pod. What about pod color? Even the pods could be different. They could be yellow or green. The flowers, what if they grow on the top versus the bottom? Or what if the plant itself is tall versus short? And so he studied 7 different characteristics and like having 2 versions of each. And again, he found this pattern. It was basically almost the same pattern of like having like 3 or 75% of the total offspring have this one trait, and then a smaller portion of the offspring having this other trait. And it was the same pattern. And so he was like, this is something. This isn't just random, and it's not just the color of the flowers. It's other traits too. So there must be some kind of pattern to how things get passed on. So by looking at these different characteristics and seeing like what showed up in the F1 generation and then what came back in the F2 generation, he came up with what he calls the law of dominance. So he looked at results for thousands of plants. And again, he was like, well, this isn't random. This is like a pattern. So he concluded that you can kind of divide the different forms of a trait into dominant and recessive. So say like the purple flowers, flower color, there's 2 versions, right? There's the purple or the white color flowers. And so he said that one of them we could call dominant and one of them we could call recessive. And so the dominant one is a trait that masks the expression of another trait if they're both present in an individual. So like the purple and white flowers in the first generation, they only showed purple, so he called the purple dominant, or the purple flower color dominant. And then the one that shows up again in the next generation that was like hidden, he called those traits recessive. So that's a trait whose expression is masked by another trait. It's like hidden by the other trait if they're both present. So if, you know, in the F1 generation, he saw that there was purple, he figured there must still be the white trait in there. It just gets masked by the purple. So he said the purple is dominant. It like bullies the other one into being the only thing that gets expressed in that case. And then the recessive trait is the one that's kind of like passive. Like, you know what? Like, fine, you be expressed. Whatever. So, yeah, for flower color in the peas, the purple colored flowers was dominant and then the white colored flowers was recessive. So the fact that the recessive trait came back and like reappeared meant that the traits were separate things. Like, they didn't get blended. It just couldn't be allowed to be expressed until the next generation. So this was because the plants had two copies of that trait. One for the purple color, one for the white color, and then the white was there. It was just hidden by the purple. So each parent gives one of their two copies to the offspring, and then they come together. So from these results, he deduced that each individual has two copies of a certain characteristic that are passed on individually to offspring. And now we know that, like, he didn't know what to call them. He just called them, like, units or discrete units of inheritance. But now we know that those two copies are genes. So the genes are, you know, an instruction for this one trait. And they get carried on chromosomes because it's chromosomes that get passed on. So we can have, you know, more terms in biology. There's always more and more terms. There's terms for everything, but it is kind of like, it makes sense. Why? Because once you have a term for a situation, it's easier to refer to that without saying, like, you know, oh, it's when this blah 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 every time. So anyway. What we call the phenotype is what we observe, the observable traits of an organism. So like the purple flower versus the white flower, that's its phenotype. What like it shows in a photograph is what how they taught me. Versus the genotype is the genetic makeup, like hidden behind the scenes, whether it has like the dominant or recessive version and how many copies of each. So we'll kind of go into that on the next slide. So again, the phenotype is like what the observable trait. So for example, in pea color, yellow versus green peas. But the genotype is like what we could call, you know, does it have two copies of the recessive or two copies of the dominant or one of each? And again, there's going to be more terms to describe each of those, but the genotype is focusing on that, the genes. And so the ratios he found, now we know, is because of certain things. So here are some more terms. Sorry. So again, it is helpful to have a term for each scenario, so you don't have to say, oh, it's the one that has two copies of the recessive one every time. So homozygous is the word we would give if there are two identical copies of one version of the trait. So for example, if we know that the organism has two copies of the dominant trait, we would say it's homozygous. So as a term, you know, the prefix homo means same. And so homozygous means having two of the exact same version of that trait. So either having both copies of the dominant trait or both copies of the recessive trait. Heterozygous, the prefix hetero means different. So heterozygous means having two different versions of the gene. So for the purple and white trait, that first generation of purple flowers were heterozygous. They did have the two different versions of the flower color, you know, behind the scenes. There was a version of the purple and a version of the white, but because the purple was dominant, that's what got expressed. And so that term is, heterozygous means something that has the two different versions of a gene. Yeah. And so phenotype is just describing like what shows the observable trait, what shows in a photograph. So either purple or white flowers or yellow or green seeds. The genotype is describing like, do you have the two versions of the recessive gene? In that case, you're homozygous recessive, two copies of the same being the recessive version, or does it have two copies of the dominant? So that's homozygous dominant. Or is it heterozygous? So yeah, these traits and these terms, we can observe the genotype, which is the genes and the letters basically. So we give a letter now to help kind of keep track of one trait. So even though we're talking about like, for example, the color of the pea pod or the pea, if we say, okay, to help us track this trait, we'll give it the letter Y. So it's still the same letter, Y, but there's two different versions. The uppercase and then the lowercase. So the uppercase is to show the dominant version, and then the lowercase is to show the recessive version. And so that kind of helps us be able to track, like, okay, these are the different versions of the traits that something has. So you can have two copies of the recessive, little y, or two copies of the dominant, big Y, big Y. And then, so if you were to cross these, kind of, the same pattern as the purple versus white flower, if you cross it, all of the first generation is going to be, it's going to show the dominant phenotype, so yellow peas. But when we cross them with the F2 generation, then we see a mix of yellow peas and then some green peas. So that means this first generation, the F1, must be heterozygous. So it must have one copy of the dominant version and one copy of the recessive version, because it comes back in the next generation. And so by, applying all these terms and like tracking the different generations, we can follow this pattern that happens over and over again with all these different traits that he looked at. So if he were to do this with the green peas versus white or yellow peas, he would see the kind of the same ratio of like 75% the yellow in the F2, and then all of a sudden some come back. And so that's 25% green. And so he, again, with all these patterns and like thousands of peas and plants, he saw this pattern. So this is, again, kind of just showing, what the difference is between the different scenarios. So if you have two copies of the dominant, that's homozygous dominant. And the genotype, we would say, big Y, big Y. But the phenotype, it would just show as yellow, because yellow is dominant, and so that's what's going to be expressed. In this scenario where the pea is heterozygous, it has one of each, one dominant and one recessive, so big Y, little y. And because the big Y is dominant, so yellow is dominant, so even if there's only one copy, that's all it takes. If there's even one copy, it's going to kind of like be dominant. It's going to be like, we're going to be expressed. And then that pea will be yellow. Versus homozygous recessive, if there's two recessive traits, that's the only way it's kind of like free to be expressed. So if you have two of the recessive versions, then it can be expressed because it's not hidden by the dominant one. So then those peas are green. And so that's why it is kind of important to be able to talk about both the genotype and the phenotype, because even though some of the peas are yellow, they could have different genotypes. So in these two scenarios, you could get yellow peas, but one of them is homozygous dominant and one of them is heterozygous. So yeah, it doesn't always, what am I trying to say? Yeah, the phenotype can look similar, but the genotype can be different. So here's an example of a trait that is recessive. So the allele, so we call the different versions of a gene alleles. So the allele for albinism, expressed here in humans, is recessive. So both of the child's parents had to be carriers to be able to have the child show that recessive allele. So like with the green, both parents had to have at least, both parents, had to have a copy of the recessive for an offspring to have both copies of the recessive and express that recessive trait. So if you have, again, even one copy of the, like, quote-unquote normal trait, then that's what gets expressed. But if you have both copies of something that's recessive, then that's the only time it'll show up, if that makes sense. So there's some, like, genetic diseases like hemophilia that is recessive, and that will only show up if the person has two copies of the recessive version. And so now we know, again, like, Mendel didn't know all this stuff, but now we know all the stuff that we do about meiosis and chromosomes and genes and DNA. And so now we know this is because of meiosis that things get divided up, and that's how you end up with one copy from each parent, because the gametes had one copy of each chromosome. So when they come together, that's how you end up with one copy of each. And so this picture is kind of showing how, again, that happens. So through meiosis, your cells, or I don't know, your parent cells, you have two copies of each chromosome. So say we're watching some trait for seed shape, and we say it's big R, little r. So again, there's still R, but there's two different versions of R, the dominant and then the recessive. And so meiosis happens, and at the end of meiosis, you have different gametes with different possible versions to give. So if this person has one copy of each, so it's heterozygous, one dominant and one recessive version of this trait, then its offspring can get one of those two chromosomes in one of the gametes. And so again, that was, meiosis. And so this ties into genetics and how we can visualize the crosses that happen with these things called Punnett squares. So Punnett squares are visual representations of, like, crossing two individuals. So when he was crossing the pollen is kind of like the sperm of plants. And he was putting them on basically like the egg cell of the other plant. And so he was doing these crosses. So Punnett squares are visualizations of that. So each parent gives one gamete. And then when they come together in fertilization, then there can be different possible combinations. So that's what a Punnett square is. It's a visual representation of crossing these two individuals and their possible gametes. And so when you set up a Punnett square, what you're setting up on, like, the top is the possible gametes from one parent. And on the other side are the possible gametes from the other parent. And then the middle, when you fill in the genotypes, is like the possible combination. So I feel like that's better shown here. So you're taking basically the genotype and tracking the possible gametes and the possible combinations. So from one flower, if this flower is heterozygous, say, seed color, is that what it says over here? So let's say, seed shape. So if the seed is like smooth, which is dominant, versus wrinkled, recessive. So if this plant has heterozygous, it's smooth shape seeds, that means it must have one copy of each. because you did something to tell. And if you take one and cross it with another one of the same kind, it's also heterozygous. Here's how we can visualize what the potential offspring might have or be made up of. So on the inside, you're kind of tracking that combination of like, okay, if this gamete has the dominant version and this gamete has the dominant version, when they get fertilized, this offspring is going to have 2 copies of the dominant version. And here's a different scenario. So say this chromosome is what gets passed on in this gamete, the lowercase r, so the recessive trait, and this gamete gets fertilized by this one, then this offspring is going to have this combination instead. It's going to get big R and little r. So it's showing like the different possible fertilizations that can happen. And this is what helped figure out like, oh, this is that pattern that Mendel saw. So for every 3 dominant, there was also 1 recessive, and it's because of this. By visualizing this, we could see, oh, okay, this is the different possible outcomes, and it's 3 to 1, that ratio that he was seeing. And that's because if there's even 1 dominant version, then remember that's what's going to be expressed. So this would be 3 smooth P's and then 1 wrinkled one, and that's that pattern that he saw. So another way to kind of visualize this is with like eye color in humans. So in humans, remember the phenotype is what shows. So you can have brown eyes or blue eyes if we're tracking, you know, just eye color. And yeah, there's more than that, but let's just right here. So brown eyes is dominant over blue eyes. So you can carry the trait for blue eyes, but if you have even 1 copy of the brown eyes, then that's what's going to be expressed. It's dominant. It's like, hey, I'm going to be expressed. So if you have a parent that has brown eyes, but her genotype is heterozygous, so she has a dominant version and a recessive version, and if the father is heterozygous also, he has brown eyes, but also has a copy of the blue eyes, then this is the different possible children they can have. Basically, like 3 of them, there's a 75% chance that they'll have brown eyes, and then there is a 25% chance that they'll have blue eyes because of the way the genes and the chromosomes get passed on and fertilization. This is, yeah, showing the different ways that like this mom can pass on one of the chromosomes, the dad could pass on 1, and in the offspring, that's how they get 1 of each. And so if they get 2 copies of the dominant trait, then that's for sure what's going to be expressed. They're homozygous dominant. If they get 1 of each, the dominant big B and then the recessive little b, then because brown big B is dominant, it's going to win. It's going to be expressed. So there will be a chance of 2 of the kids having brown eyes, but they're heterozygous genotype. And then there's a 25% chance that this child happens to inherit both copies of the recessive gene. And so that's why this child has blue eyes. So that can happen in families. You might see like a lot of family members and most of the generations have brown eyes, and then sometimes like a trait like blue eyes or green eyes can come back in a generation because of this possibility of what gets passed on. So yeah, you could happen to have both copies be dominant, or you can have 1 copy that's dominant and 1's recessive. So you're heterozygous and you could be like carrying that trait. And it's just kind of like a matter of chance, basically, like what gamete has what and what gamete fertilizes the other one, basically. So this isn't like a hard and fast rule that like if you both have brown eyes and you happen to be heterozygous, it's not for sure. Like every 3 of your kids will have, if you have 3 kids, they should all have brown eyes, and then the 4th kid will have blue eyes. It's just like a chance, basically. So there's a 75% chance that one of your kids can either have brown eyes or the blue eyes. And each child is, again, it's a chance, if that makes sense. Yeah. So don't, if you have brown eyes, don't specifically go out and have kids with someone that has blue eyes just because you want your kid to have blue eyes, because it's not guaranteed, is what I'm kind of trying to say. It's all up to chance about the chromosomes that get passed on and the gametes that fertilize each other. Yeah. So this is kind of saying something that we already covered. And so anytime you're crossing an individual with another one and you're following one trait, so basically what Mendel did was called a monohybrid cross. So for each of those different traits, he was following 2 different versions, but he made kind of like a hybrid, so that's why it's called a monohybrid cross. He's only looking at one trait, mono, and then he's crossing specifically one with another one, so he's kind of making hybrids. So yeah, when you're looking at one trait, you are doing what's called a monohybrid cross. So when he looked at seed color, yellow versus green, he did a cross, and the F1, again, now we know is heterozygous. It had a dominant version and a recessive version. And that's because of the gametes. So this gamete can only really give a dominant version because that's all there is. This gamete would only for sure have a recessive version because that's all it has. And so this combination is heterozygous. And so when this F1 decides, well, not decides, but when this F1 has gametes, it can either give the dominant version or the recessive version because it has one of each. So when you're doing a monohybrid cross and you do a Punnett square, this is kind of what you're trying to track. Like, okay, this is what the parent has, and so this is what the gametes can be or give. And then if this gamete gets fertilized by this gamete, then this is the outcome. If this gamete gets fertilized by this one, then this is the outcome. So maybe, hopefully, this kind of demonstrates all the concepts together in one grand slam. Yeah, and now we know that this explains the ratios he was finding because of the different genotypes that produce phenotypes, and this all allows you to track that. So say you find a pea on the floor and it's yellow, and you don't know if it's homozygous dominant, big Y, or if it's heterozygous, big Y, little y, because either one can give that yellow color. So if you wanted to figure out what something is, you would do what's called a test cross. So a test cross can be performed to see whether an organism expressing a dominant trait, is it homozygous dominant or is it heterozygous? So if you have a yellow pea, you know for sure it has at least one copy of the dominant version because it's yellow. But you don't know if it's, again, big Y, or big Y, little y. So what you do is cross it with one that's all recessive. And so when you cross it with something that's all recessive, depending on what shows up in the next generation, tells you what the parent was. So for example, say you cross these two, so your yellow pea with the green pea, it could go one of two ways. Either all the offspring are yellow, and so what that tells you is that parent yellow pea was big Y, because all the offspring would have had to inherit a copy of the recessive from one of the parents, recessive from one of the parents, and then a copy of the dominant from the other parent. So if all of them have big Y, then that means it must have had 2 big Ys to give. But say you do this cross and you find it goes half and half, so half of the peas are green and half of the peas are yellow. That means that yellow pea had to have a recessive version because that's the only way green gets expressed if there's 2 copies of recessive. So that means this one had to be big Y, little y. So again, in this example, which isn't, one of my favorite examples is this wrestler, Daniel Bryan, and his wife, who was also a wrestler, Brie Bella. And so this is such a tangent, but so Daniel Bryan has blue eyes and his wife Brie has brown eyes. And so because, she has brown eyes, there's at least 1 copy of the dominant version. And so this would be a different cross, but this is just to show, again, like brown eyes is dominant versus blue eyes. And so when they had kids, they've had 2 kids, and both kids have blue eyes. That means she must have been a carrier. Because if he had blue eyes, he would be big B or little b. But if she has brown eyes and any of the kids have blue eyes, then that means she is heterozygous, because she had to have also given a little b. If that makes sense. Hopefully. Again, I feel like it's always more relevant to think about, like, how does it apply to us and humans and daily life and our genes. So that would kind of be called, that's the concept of a test cross. You cross, if you don't know what the trait is that has a dominant version in an organism, like a pea plant, you would do a test cross. And you would cross it with something that's all recessive to see what that parent was. And like I said, you probably wouldn't purposely do this with humans, but it was just a neat example of something like that happening. And so, what Mendel did next was he was tracking, just 1 trait at a time, but he was like, I wonder what would happen if you tracked 2 different traits at a time. He just wanted to make life harder for himself. So he studied 2 traits at once in different crosses. So both seed color, yellow versus green, and then seed shape, smooth versus wrinkled. He was like, I'm going to track both of those things when I do a cross and then see what the offspring gives. And so when he crossed a yellow smooth seed with a green wrinkled seed, at first, the first generation was all yellow smooth. So again, he was like, okay, so the dominant version of each thing is winning, and that's what's being expressed. And then he crossed one with another one. And he was like, what the heck? This is like a huge combination, like differences in combination than I thought would happen. So he thought maybe they'd mostly all be like 75% yellow and smooth, because that was the ratio that he found in the other one, right? But no, he found a different mix. He was like, whoa. I thought all the yellows were going to be smooth, but there are some yellows that are wrinkled. And there are some green that are smooth, and there's some green that are wrinkled, but like he saw these new combinations that he maybe didn't expect before, and he was like, whoa. Okay, so that means that each trait gets separated differently. Like yellow and smooth is not tied together, even though it's kind of more common, it's not tied, because there are some that are yellow and wrinkled, and there are some that are green that are smooth. So then these 2 things get passed on differently, separately. And so what this is called is a dihybrid cross. So again, he's following 2 different traits in hybridizing plants. So it's a dihybrid cross, 2. You guys, I'm not going to expect you guys to learn how to do this. It kind of helps because you can kind of track it through, but this is just showing this concept. So Mendel came up with what he called his like 2 laws. Again, like everything has to have like a name and a law and whatever. So he came up with these 2 concepts basically. So the law of segregation says that the different versions of a trait get separated equally into gametes. So there's an equal likelihood of inheriting any combination of genes. So for example, the yellow versus green pea color, he said the paired genes separate equally. So 50% are going to have the, this is a different concept. So 50% are going to get one copy, and then 50% are going to get the other copy. So again, I don't know why everything has to have its own, like, name and whatever. He's just stating the concept that the different versions of a trait get passed on. And that's it. So if you have, like, 2 versions of a trait, then they're just going to get separated. And that's basically it. And then the other thing, the concept that he stated, was called the law of independent assortment. And so that says that genes don't influence each other with regard to sorting alleles into gametes. So even if you're tracking 2 different genes and 2 different traits, they're not going to be tied together. Like yellow is not always going to give smooth also. So each thing assorts independently. So for example, during meiosis, now that we've covered meiosis, it's possible that the chromosomes line up this way, so the blue's on one side and then the red's on that side. But it's also possible that it lines up in the opposite way. So one blue on this side and one red, and one red on this side and one blue. And so those 2 different traits that he's following, like yellow versus green and wrinkled versus smooth, they're 2 different things, and they could be on 2 different chromosomes. So they're not going to be tied together. It's not always going to be carried around together. They assort independently. So yeah, these are just some of the concepts that he studied and, like, came up with. And so now we know that things can be more complicated. Again, he got really lucky. He happened to study traits with only one mode of inheritance that wasn't that complicated. It was just dominant versus recessive. So he was really lucky in that way. So that was really good to know. But afterwards, we learned, oh, there's other things that does not follow those specific patterns. Like there's different scenarios where some traits do blend or they mix in a weird way than what we thought before. So there are some models of inheritance that don't follow dominant versus recessive super completely. So there is what's called incomplete dominance. So say red and a white flower, and you cross them and it's pink, that's totally different. It's a different model because in this case, like, the 2 things are getting blended. There's a mixture of the 2 different versions. So that is called incomplete dominance. Like, one is dominant, but it's not completely dominant. It doesn't completely win out. Like, some of it does get expressed. So there's a mixture. So red is kind of dominant, but it's not complete dominance. So it's just a blend of the 2 different traits. Codominance is a different version where it's like they're both dominant at the same time. Like they're fighting, but they kind of like both end up getting expressed, and so they're both co-dominant. So there's going to be parts of the flower that are red and parts that are white. So that's, different because it's not a blend specifically. It's just like they're both being expressed. In the original form, they're both trying to be dominant. So again, he got lucky studying stuff that was just the model of complete dominance. It's like this or this, because there are some other flowers that would have shown different stuff happening. Yeah, so this is another example of incomplete dominance. This happens in this other flower, the snapdragon, and so it will have pink flowers. So that's a model of incomplete dominance. And then an example of a real-life model of codominance is blood types. So this is kind of complicated, but kind of not. So, you know, we can be either type A or type B, or you could be both, type AB blood. And so that is an exhibit of like codominance. So if you have A and B, that's technically codominance. They're both being expressed. So you could be type A or type B, or A and B. AB is codominance because they're both being expressed. But then it does get more complicated because there's even more alleles. So you could be type A, type AB, or type B. Or there's a third version where it is type O. So in that case, you don't get either A or B, and so that's how you get type O blood, which actually I am. So yeah, stuff can be a lot more complicated than what he was studying. So this is how you would get type A or type AB, because you're getting either two versions of the A, or two versions of the B, or one version of each, or none of those versions, and that's how you end up type O. And then there are some traits that are tied, you know, again, more complicated. So there are some traits that are tied to like sex. So there are chromosomes that are called the sex chromosomes. And so female organisms have 2 copies of the X chromosomes, and males have an X and a Y. And so some traits are tied to the X chromosome. And so if you happen to have just one version on the X and you're male, then that means you only have that one version, if that makes sense. So this is kind of how we show that. We add it as like a little, almost like an exponent. It's called a superscript. So we would put the letters on that if it's on the X chromosome, and that's how we would track it through different generations. Yeah, so some traits are sex-linked, and for example, color blindness in humans is sex-linked, and so males happen to be colorblind more often because if they get even one copy of the recessive on the X, then that's all they can express, if that makes sense. To be honest, I don't remember what the slide is doing here. I don't remember, so just ignore that. And again, to have things even more complex, there is this mode of inheritance called epistasis, and that's with when one gene kind of controls expression of a different gene, and not in like a dominant way. It's one gene controlling a totally different gene. So one of the genes can have the different alleles, you know, this color or this color, but that other gene can control whether or not there's any color put in at all. So in a lot of cases, a lot of traits actually have epistasis, so that like observable characteristics are actually under the control of multiple genes acting together. So here's an example in mice, but it's also actually relevant in Labrador Retriever dogs, so in labs. They also have this same kind of model of epistasis. So the coat color gene says, okay, you can either be brown or black. And this other gene will say, actually, I decide whether or not you put any color at all. And so you can have, you know, either brown or black, or, you know, both copies or whatever. But the other gene will control if there's any color at all. So some can be, if you're following the 2 different genes in a dihybrid cross, you would see that some can be brown, some can be black, but some will be white because that other gene is saying, no, you're not putting any color in at all. So that's actually how you get golden retrievers. That's technically the like absence of color. It's kind of more clear here, and obviously, you know, like an albino mouse. That other gene is saying, hey, you're not going to put any color at all. I don't care what color it says, you're not putting any color in at all. And so that's why some of them are albino mice. Even if they do carry either the black or the brown, the other gene is saying like, no, you're not putting anything. So yeah, that's how you can have like chocolate labs and black labs, but also golden labs. That's technically the no color option. Yeah, so that's epistasis. When two genes are involved, one is just controlling the other gene. So like I said, Mendel got really lucky studying this stuff that he did because it didn't follow all this crazy stuff. But it was really good in leading us to know, okay, something is passed on to offspring in a kind of pattern, and now we know it's chromosomes, and we can build off what he knows now. So, hopefully this all makes sense now. So hopefully you can kind of explain how Mendel's experiments were successful because of like those different traits. Recognize basically the outcomes of a monohybrid cross involving dominant and recessive things. Explain the relationship between genotypes and phenotypes. Use a Punnett square to kind of map, you know, the genotypes and phenotypes in a monohybrid cross. So don't worry about dihybrid, but kind of get like how you do a monohybrid cross. Explain Mendel's law of segregation and independent assortment, and then the purpose and methods of a test cross. So hopefully that all seems not too bad, but again, there's always additional resources to watch and kind of see things play out, especially if you're a visual learner or like even an auditory learner, like hearing it explained a different way is super helpful. Yeah, so that is lecture 7. Oh God.